Hello, everyone, and welcome back to our summit today. Our first presentation today is going to be by Melissa Thompson, who's a wildlife biologist that works for Fish and Wildlife Services. And we also have Blake McCann with her. He is the Chief of Resource Management at Theodore Roosevelt National Park. So we're happy that both of them are here, and they're going to share a presentation with some research that Melissa has done. So again, we just remind everybody you are muted and your videos are off. If you have any questions, please put them in the chat and they will be happy to have a discussion after Melissa's presentation. So thank you again, you guys, for being here. We really appreciate it.
Thank you, Chris. Melissa, will you please take it away?
Let me share my screen here. All right. Quick introduction of myself. My name is Melissa Thompson. I worked for 8 years at Theodore Roosevelt National Park, working with the wildlife there, with the free-roaming horses and with a bunch of the other native species, particularly the bison and the elk. And then I had the opportunity to go to graduate school at the University of North Dakota to get a master's degree. My research project for my thesis was on the genetics of the horses of Theodore Roosevelt National Park. So that's what I'm going to be presenting on here. My project on the population genomics, looking into ancestral relationships of the park herd. So first of all, I just want to recognize my advisors at UND. Dr. Turkren and Dr. Becky Simmons were very helpful. And then also Dr. Blake McCann with Theodore Roosevelt, who his expertise was very helpful to the project and also helped provide funding through the National Park Service for the genetic analyses. So on a quick introduction to Theodore Roosevelt National Park, it's located in the western North Dakota Badlands, and it is home to many different species of animals, in particular many large herbivores, such as bison, mule deer, white-tailed deer, elk, moose, pronghorn, bighorn sheep, and a herd of free-roaming horses, which I'll be talking about today. So all of these species share space within the park. The park is entirely fenced in to contain the bison and horses. So since there's limited space, the populations need to be managed in a way to maintain healthy habitat for them all. One of the questions that comes up a lot in discussion about management of the horses in particular is about the origin of the herd and those genetic relationships to other horses or other horse breeds. So for my study, what I was looking at, I had a couple questions. What horse breeds are these horses related to? And knowing that, can we learn more about the origin of the herd? First, a brief history of horses. So horses evolved in North America in many different species, some of which spread across the Bering Land Bridge into Eurasia, where they were eventually domesticated. All of the horse species left in the Americas went extinct around the beginning of the Holocene, but horses were later reintroduced into Americas by first the Spanish explorers and then by other European colonists. Horse use was adopted by many native peoples who developed rich horse cultures and contributed to the spread of domestic horses throughout the Americas. In North Dakota specifically, there were a few different types of horses in use in the late 1800s to early 1900s. The horses of the American Indians tended to have Spanish blood from that initial reintroduction of horses into the Americas. And then the ranchers moving into North Dakota brought with them mostly English breeds of horses from the East Coast. The largest horse ranch in the entire region bred Percherons, a type of draft horse. In addition, horses which could be considered early quarter horses were brought up from Texas along the cattle drive trails. The quarter horse is an American breed which was developed by crossing the Spanish type horses with English breeds and then later crossing them again with thoroughbreds. So there was a lot of interbreeding between types, trying to breed the best ranch horses. And you can see an example of that in this picture from the Hydacopa Ranch of this Percheron stallion here, who was turned out with a bunch of mares of other types. So then what we know about the history of the TRNP herd specifically is that they are descendants of free-roaming horses and mules that were, well, not descendants of the mules, free-roaming horses that were present before the park was fenced. Oftentimes ranchers would run their stock loose on open range to forage and breed on their own, and then they'd go and recapture them when they had need. So when the park fence was built in 1954, they attempted to remove all of the horses from within the park, since most of them were branded and had owners. They were unsuccessful in capturing all of the horses, though, and some continued to elude capture over the next 10 years. The best count of remaining individuals that we have states that there were 16 horses in the park in 1965. The local oral history suggests that the horses which eluded capture were the wildest and had the most Spanish blood. In the 1970s, the park decided to keep the horses as a demonstration herd and moved to conducting roundups as population management when the herd grew too large. There have been 10 formal roundups conducted since then, where each time the population was reduced by about half. This means genetically that the TRNP herd has effectively undergone 11 genetic bottleneck events, where a major reduction in population size can mean less genetic diversity afterwards, just based on which individuals are left. Since then, the herd has mostly been a closed population, with the main exception of a handful of stallions, which were introduced in the 1980s. They had varying levels of reproductive success, except there was one, a Shire paint cross, who was very successful. He became the most dominant band stallion in the park for about a decade and sired many offspring. Though he and many of his descendants were later removed. So based on that known history, I hypothesized that the breeds which would be most similar to the TRNP horses were the Spanish type breeds, or the quarter horse or American paint horse, which are by far the most popular ranch horses today, or draft breeds like the Shire or Percheron. So for the methods, I'm going to summarize the methods fairly quickly since there's a lot of complicated genetics terminology that you don't really need to know. But first step was to collect the DNA. So the park already had hair samples from every horse that came through the 2013 roundup. So I had access to hair samples for the older horses in the herd, but I also used biopsy darting to collect tissue samples from a few additional horses. In total, I selected 118 samples for genotyping, which represented 98% of the herd at that time, if you include the offspring of the sampled individuals. Then those DNA samples were genotyped for what are called SNPs using a commercially available SNP array. But what are SNPs? SNPs are single nucleotide polymorphisms, where a single nucleotide along the DNA sequence is different in some individuals than it is in others. These are mostly located in non-coding regions, meaning that they don't change any genes and there isn't any active selection happening on them, so they're really useful for population genomics. Since the entire horse genome has been mapped, we know where millions of those SNPs are located. And so we can use much higher numbers of markers for comparison between populations than we could in the past. Can use easily tens to hundreds of thousands of markers. So for my work, I combined the TRNP horse genotypes with a published data set of SNP genotypes from horses of 35 different breeds that were genotyped by Pedersen et al. After quality control filtering on the combined data set, I had over 38,000 SNPs to work with. Then I ran those genotypes through a bunch of different population genomics analyses, such as a principal components analysis, FST computation, a Bayesian cluster analysis, phylogenetic analysis, and a runs of homozygosity analysis. These are all different ways of looking at population genomics, and by comparing them, you can kind of pick out patterns and draw better conclusions. So, on to the results of those analyses. First off, this is the plot from the principal components analysis, or PCA. This program tries to simplify the variation in the broader data set into what are called principal components, or vectors that give the genetic variance in the data set a value that can be plotted along a line. So PC1, principal component 1, is the line that the program found to represent the most variation in the data set and is the x-axis of this plot. And then PC2 represents the second highest description of variance and is the y-axis. Each of these points out in the plot represent where one individual horse fell on both of those axes. Here, we'll start looking at this one on PC1 on one side are the thoroughbreds, and then separated on the other side of PC1 are the big draft breeds and the mini ponies, which have been under artificial selection for their size. Principal component 2, the y-axis there, shows the divergence of the draft breeds away from the mini pony breeds. The TRNP horses are right here in pink in the middle of the plot. And I've circled some of the other breeds of interest here too. First off, the Spanish breeds, all 5 of the Spanish breeds in this data set are clustered right on top of each other here. The Shires are out in the draft branch here, and the quarter horses are here. The quarter horses are a really good example of what crossbreeding and admixture look like on a PCA plot, where their recent crossbreeding with thoroughbreds and the rest of the horses here has left them kind of strung out in a line on the plot. So looking back at the TRNP horses, you can see they don't form as tight a cluster as some of the other breeds, probably due to admixture in their history. And they seem to be kind of separated out from the other breeds, kind of in the direction of the draft branch here. Next, we'll look at the FST matrix quickly. So FST is a measure of population differentiation based on the difference in allele frequencies between each pair of breeds. High FST means that there is more difference between the pair. The thing to point out here is that the TRNP horses have a mostly higher than average difference to the other breeds. but they have a lower difference to the quarter horses than to some of the Spanish breeds in green down along here. On to the Bayesian clustering. So this program tries to reverse engineer the genetics and calculate a number of ancestral clusters that contributed genetics to the present day population. According to the program, the number of ancestral clusters that best describes the current variation is 25. So there are 25 colors shown in this graph. Each vertical line in the graph represents one individual's genome, with the colors showing what proportion of their genome was assigned to each ancestral cluster. So in each of the breeds, lined up by breed here along the bottom, most of the individuals had generally the same color within the breed. Some breeds have more mixed ancestry, according to this, like the paint and quarter horse here. But the TRNP horses are assigned to their own ancestral cluster, this kind of red one color, which only had a little bit of other colors from any of the other clusters. Also, none of the other breeds here really had much of that color show up in their portions. So it's a pretty distinct cluster from this. And then this is the phylogenetic tree. First off, thing to point out, see how short these internal branches are here compared to the long external branches? That's pretty common for domestic species that have undergone rapid differentiation, usually due to artificial selection for whatever traits were desired. The tiny little numbers in the middle are the confidence values, which shows how confident the program was that that junction was accurate. Most of the internal junctions don't have high enough confidence values to report, in large part because this phylogenetic analysis can't incorporate the crossbreeding between breeds that is so common. It's still interesting, though, to see where the TRNP horses were placed. They are this branch right here, which was placed here in amongst all of these draft breeds, like the Shire, Clydesdale, Phil Pony, Billigen, and Percheron. We don't have significant confidence in that branch. However, there is one branch with high confidence that is particularly relevant, and that is this one right here. This one with 91% confidence separates all 5 of the Spanish breeds away from the rest. So because of this separation, since the TRNP herd wasn't placed with this branch, it's unlikely that there is a strong Spanish ancestry for the TRNP horses. And then finally, one of my favorites. So one way of estimating inbreeding is to use runs of homozygosity, which are stretches of the chromosome where all of the SNPs are homozygous, meaning that they have the same allele from each parent. If there is a long stretch of homozygous markers along a chromosome, those parents likely each got that version of the chromosome from a common ancestor somewhere in their pedigree. This program that I used identifies these runs and then assigns them to generation classes by length. Because these stretches can be broken up by recombination during meiosis, which is the cell division process to create eggs and sperm, a long run of homozygosity is less likely to be inherited from a more recent ancestor, or a long run is more likely to be inherited from a more recent ancestor, since there were less chances for recombination. And a shorter run is more likely to be inherited from a more distant ancestor. So this graph shows the proportion of the genome that is made up of runs of homozygosity for a random selection of 30 of the park horses with different colors to represent the generation classes. You can see some of the individuals have as much as 40% of their genome that is homozygous. And then looking at the colors in the graph, you can see most of the runs are in the red and orange class, which corresponds to a common ancestor about 4 to 8 generations ago. So freely breeding horses have a genetic generation time of about 10 years. So multiplying that out, these common ancestors of the herd are from about 40 to 80 years ago. And that overlaps with 60 to 70 years ago, which is when the park was fenced and when there was that substantial bottleneck that reduced the population to 16 individuals. This suggests that the main impact of inbreeding was probably from that initial bottleneck, although we do see some evidence of more recent inbreeding here with these darker brown segments. So now we've done the analyses. What conclusions can we draw from all of it? Pulling together all the results, there were 2 main conclusions that I made. Firstly, that the TRNP herd is distinct on the population level, though not necessarily on an individual level, but that is most likely because they are a small isolated population that is only becoming more and more like each other with each generation, and not necessarily because they have a different origin than any other horse breed. Some of the genetic mechanisms of evolution that are at play here are the population bottlenecks that I've mentioned, where a major reduction in population size can end up with allele loss. There's isolation, where there is no gene flow and there are no new alleles being introduced. Inbreeding, where the offspring in subsequent generations are becoming more homozygous. And lastly, genetic drift, which is just a random loss of alleles just based on who may or may not have any offspring and which alleles they may or may not pass on to those offspring. Second conclusion, as far as the breed relationships, so no one of these analyses gives us a really obvious ancestral relationship for the TRNP horses, but there are some patterns across the analyses. So the horses have an admixed origin with multiple sources, but their strongest relationship is with the draft breeds. This is probably at least partly due to the Shire Stallion that was introduced in the 80s, but it may also reflect earlier Percheron breeding in the area. However, there is not much genetic evidence for a primarily Spanish origin for these horses. So to sum it up, the TRNP herd is a separate population with the strongest connection to draft breeds. And with that, I'll take any questions.
Thank you very much, Melissa. That was really interesting. Can you talk about the difference between inbreeding and line breeding?
Line breeding is a more artificial selection sort of thing, where human influence is particularly choosing out what lines to breed. That can result in inbreeding. Inbreeding is a more general term that describes what happens when you have multiple common ancestors. So you do, especially with some of the more specialized and specific breeds tend to see more inbreeding within that population.
Thank you. Does anybody else have any questions? Krista wants to know if the study was shared with the park. And I don't know if you want to talk about that in your relationship and
Mm-hmm. Yep. So Blake McCann is the director of resources at the park, and he was on my committee, helped out with it. So he was intimately involved with figuring this out. We published this. I published it as a thesis and then submitted it to a peer-reviewed journal. So it's published publicly, open access. So anybody can go and read the full paper.
Craig wants to know if there's any chance of allowing a larger population in a larger habitat so the danger of inbreeding can be overcome and this unique population can survive.
That's more of a management question than about the science results. So that would be Blake, do you have anything to say for that?
Sure. Yeah, there's different ways that you can get there. Obviously, introducing new genetics is one way to improve genetic diversity of a herd. A large population size really buffers things like drift, so genetic drift, that random loss of allelic diversity. And so if you think about it, if we have a rare gene that occurs in a population, let's say 1 in every 100 individuals, if you only have 100 animals in the population, then by chance, if that one animal gets struck by lightning, for instance, it's gone. Whereas if you have 500, then there's 5 copies. So that's just kind of the relationship of drift with population size. So to answer the question, it's one tool that could be applied in how you manage a population larger size, but there are others such as implementing gene flow.
How does the predominance of draft breeds present itself in the current TRMP horses?
I would ask Melissa to address that.
Yeah, like phenotypically, for phenotypically the appearance, the outside appearance of the animal, how the genes are expressed. The park horses tend to be, they're kind of on the large sides for a herd of free roaming horses, which is probably due to some of that draft breed influence that tend to be a little bit stockier than some others. So there's a little bit of phenotypic evidence of that too.
Krista wants to know, do you currently work with the herd or at the park, or will you do more work with the park on the genetics of the herd?
So I don't currently work with the park anymore. I used to work with them. I worked with the herd for 8 years. I have moved on to a different position now with the Fish and Wildlife Service. And so I'm not currently doing any work at the park, but there's a possibility that I could come back to the park again or something.
We're always happy to have Melissa help us with things. I'll just speak to say that we have other collaborators with USGS, and like in this instance, I keep calling on Melissa to come and present on her findings and her work. It was nice working with University of North Dakota. One of the things we get from academic institutions, in addition to other scientists within DOI, is just additional perspective. And certainly we've been very lucky with the committee members Melissa had there, but also with cooperation across other universities in ongoing work such as Texas A&M in dealing with answering questions about horse genetics.
Can one of you send me the link to your full paper? Because then I can just put it on the website so that people are asking where they can read your study. So if you want to just email that to me later, I can add it to the website and then people can access it from there.
Too. So it was published in Ecology and Evolution. Here's the DOI. Let me see if I can find the chat. Okay, so that should be the link to the paper. And then I can also send it to you by email, Chris.
I copied it too. I can just add it to the website also so that people have that information. Does anybody have any other questions for Melissa or Blake? People are saying it was a very fascinating presentation, so thank you very much. We appreciate you guys doing this and for all your research. All right. Well, thank you guys for being here and for all the work that you guys did. Everybody's saying it was really fascinating research, so thank you again for doing that and for being part of our summit this year.
Thank you, Chris. And we'll look forward to an opportunity again to participate.
Thank you very much.
Thank you.
Thank you, everybody. We'll see you at 2 o'clock for our next presentation.